Welcome. This is Sacramento City College Biology 440 General Microbiology Lab. This video is going to cover information in Chapter 2 of our lab manual, lab equipment and media. The first topic we're going to discuss is how we grow our microbes. Mostly we'll be working with bacteria in our lab. So we want to discuss how will we encourage our bacteria to grow and divide. And of course, they, like us, require food, nutrients. We refer to the food that we provide our microbes as media, is plural, medium is singular. And media is classified according to which ingredients are present, whether it's a solid, semi-solid, or solid. So we'll start out with a discussion of media based on whether they're liquids or solids. So the first type of media then, a liquid media, would be referred to as a broth. And broth, we can think of like chicken broth or vegetable broth, almost like a soup. You can see that this broth, which is tryptic soy broth, is transparent. It's not cloudy. It's not turbid, suggesting that this broth has probably been autoclaved, destroying all cellular organisms. And thus, we're hopeful that it's sterile. So we could transfer microbes into this broth if they had the transport proteins to transport nutrients from the broth across the cell membrane. If they had the enzymes that they could use those nutrients, then they could grow and divide, and eventually we'd see that the broth would become turbid, suggesting that cells are present. Now, a huge safety item is that many of the tubes that we use to grow our microbes, as this one, we can see that it's a screw cap tube. So you can see the threads here. And so when we put the caps on, after we've inoculated, transfer our microbes into the cap, we want to tighten and then barely loosen. And this is crucial because we want to make sure that the gases produced while the microbes are growing don't build up within the tube. As a consequence, for example, if we tighten the cap here, our microbes grew, releasing lots of gases. The pressure inside the tube could increase, so that when we then went to work with the tube to move it, the tube might explode in our hand, almost like a little hand grenade. So we always want to remember that whenever we're growing microbes in screw cap tubes, tighten the cap and then barely loosen so the gases can vent. Now if we take our broth and if we add a solidifying agent, in microbiology the most common solidifying agent is an extract of seaweed called agar, then we end up with solid media. So here we have what's called an agar deep. It too is in a screw cap tube. We can see that as the agar solidified, the tube was placed in an upright position, so it makes this big butt of solid agar. So agar deeps can be used, for example, to see if microbes can grow anaerobically. Anaerobically means without air. What we might do is, using a needle, stab or inoculate our bacteria down into the very butt or bottom of this agar deep and then incubate it and then see if the microbes could grow anaerobically. Again, we always want to remember that when we're growing microbes using these agar deeps, we always want to barely loosen the cap so that they can vent. If, when we autoclave our tubes containing our agar, if we let the tubes solidify at an angle, we can see we get this nice ramp or slant of agar. This increases the surface area upon which we could transfer our microbes to see if they could grow aerobically with air. And again, you guys want to remember to loosen, barely loosen those screw caps if we inoculate them with our microbes. Now, previously we had discussed how with the broth appeared to be sterile, lacking any microbial growth because it didn't appear cloudy. Just as an example of here of what we mean by cloudy or turbidity is in, this is called thioglycolate media. Thioglycolate is a substance which reduces molecular oxygen, creates an anaerobic environment. We have an oxidation reduction indicator here that turns green when oxygen is present. The purpose here is I just wanted to show you the cloudiness or turbidity, telling us that cells are present. We have microbes growing in this tube. Now we have additional containers into which we can place our media, and another container would be a petri dish. Now this is a lovely reusable glass petri dish. It's important for us to note that there's two parts of the petri dish. There's a larger diameter lid and a smaller diameter bottom. All right. And with these petri dishes, what we do is, after autoclaving while the agar is still liquid, the liquid agar would be poured into the bottom of the plate, right, and then covered, and then permitted to solidify. So here we have an example of what we call an agar plate. So we can see in this case, this is a disposable plastic petri dish that's been filled with agar. So again, here we see the lid is on top, the larger diameter, and the bottom, the smaller diameter bottom, into which the agar has been poured. So we would transfer bacteria to the surface of the agar. And you'll notice here that we've taped the lid to the bottom of the plate. This is important. We don't want the lid to fall off while the bacteria are growing. So we always want to tape the lid to the bottom. You can see we have two pieces of tape here. And one piece of tape we've dog-eared. We've turned the flap over so this is easy to grab. Right? So when we want to open our plate, we always open them like a clamshell, like so. We never want to take the lid all the way off. And the reason for this is that we're going to discover there are lots of microbes even in the air, floating in the air, maybe associated with dust particles. We always want to try to prevent unwanted environmental microbes from growing in our media. So if we took the lid all the way off, we would have lots of microbes that would just fall from the air onto the surface of the plate, thus contaminating our plate. We don't want to have contamination. So to prevent contamination with airborne microbes, we always open our plates like a clamshell. And what we're hoping is, as airborne microbes fall, they're going to hit that lid and will reduce the chance of contamination of the surface of our agar. We'll transfer the microbes we want to grow to the surface of the agar plate. Then we're going to close it, tape it shut, and very importantly, when we incubate, we always want to invert the plates. And invert means we're going to turn them upside down, right? So the bottom would be facing up. And also very importantly, in microbiology is the use of labels. So a label has to have basic information. Here I've made a tape label. I'm going to put it on the bottom of my plate. That's where we always label our agar plates. And we'll see here the basic information for our label is we always want to have the microbe's name. We want to have the student's name. So here we have Louis Pasteur as our student. We put the date, and we always put our lab section. So we're pretending this is a Monday, Wednesday, 8 a.m. lab section. And again, when we incubate the plates, we always put the plate in the incubator inverted or upside down, so we always see the label pointing upwards. We've talked about inoculating or transferring microbes into our media, so we want to discuss some of the instruments we use to transfer or move our microbes from one place to the other. And two of the really common ways that we can transfer our microbes is through the use of an inoculating loop or inoculating needle. My preference is to use the inoculating loop. Let me see if I can get that loop in here. And you can see the inoculating loop. It's wire that's been formed into a loop. Okay, so this would be the part that we're going to use to transfer our bacteria or microbes from one place to the other. And then I'm going to do a no-no here. I'm going to grab the loop just so you can see the rest of the inoculating loop. You can see there's a metal handle here. Okay, so we're always going to use the loop holding the handle here. We never use a loop holding the actual loop part here. And indeed what we're going to do when we're transferring microbes, again, we want to avoid contamination with unwanted environmental microbes. So we'll always need to sterilize our loops before we transfer our bacteria. So the way we would do that is we would light our Bunsen burners, and then we're going to hold the loop in the Bunsen burner flame until it's red hot. And we'll gradually move the loop so the entire wire portion becomes red hot. That incinerates any microbe. It sterilizes the loop, right? And then we will very lightly flame the metal handle. If we held the metal handle in the flame, it would probably melt. Okay? So again, we're going to sterilize our loop by flaming. Once it's sterile, we don't want it to touch anything else. If it touches anything, we would presume it's contaminated, so we'd have to reflame it. Another inoculating instrument is an inoculating needle. And you can see very similar to the loop. It's just a straight wire. And needles are often used to do stab inoculations. You'll recall we discussed our auger deep. And let's say that we wanted to transfer some bacteria into the very bottom of our auger deep, right down here. So the way we would do that is that we would use our inoculating needle, and using sterile techniques, which we'll demonstrate to you, we'd actually stab the microbes right through that auger deep down to the very bottom, and then we would withdraw. And we'll have a lab on how to do aseptic transfers. I'll discuss in greater detail. Another way that we can transfer microbes is we can use sterile transfer pipettes, or excuse me, these may or may not be sterile. This one is not sterile, but it lets us transfer small volumes of, say, liquid media from one place to another. Or maybe we had maybe pond water, for example, that we wanted to use as a source of microbes. We could use a transfer pipette to transfer small volumes of liquids. Another way to move microbes is through the use of a glass, what's called a Pasteur pipette. We have here. Now these aren't marked with units, so this is a less accurate way of transferring fluids. But what we would do is use a plastic bulb, like so. All right. And then we could transfer, say, fluid from, again, from a liquid source, say either into maybe onto an auger plate or into, say, broth. What we want to remember is that when we're finished with our Pasteur pipettes, they never go into the regular trash. They can actually cause a lot of damage if people come along and are helping us clean up. The Pasteur pipettes can act as almost like a needle and jam right into the hand. So glass, including Pasteur pipettes, always go into the sharps or broken glass containers. We want to remember always to remove the rubber bulb, right? Because these we don't want to throw away. Another way to transfer bacteria or to transfer liquids of any time is with the use of serological pipettes. And it's really important that we're familiar with how to read units on serological pipettes. They come in different sizes, and it's important to know how to read the serological pipette. So you'll see at the blunt end at this point, it has a cotton plug there to prevent aspiration of fluids. Okay, and then there's a tapered end down here. This is the end that we would immerse into the fluid that we're going to transfer, and then you can see that there's lines, little units. Some of the units aren't numbered and others are, so we want to know what's the total volume, and then we want to know the volume of the smallest units and the volume of the larger numbered units. So for us to figure that out, we're going to go up here towards the blunt end, and you'll see that there's some numbers here. The first number, 1, tells me the total volume is 1 mil. And then the second number tells me what's the volume of the smallest units. And here we'll see that smallest units have a volume of 1/100th mil, or 0.01 mil. So we always want to make sure that we include the volume at the tip. So the volume from the very tip to this 0.9 right here, that would be 0.1 mil. The volume from the 0.9 to the 0.8, that would be 0.1 mil. So if I drew up fluid from the tip to the 0.8, that would be 0.2 mils total. The smaller units here that lack numbers, each of those is 1/100th mil. So you can see there's 10 1/100th mils in 0.1 mil. Okay. Now the total volume, as you will recall, from the very tip, if we drew up fluid all the way up to the 0 here, The total volume would be 1 mil. Okay, all right, so if we were to count the units here, 0.1 mil, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, 0.8, 0.9, or 1 mil. Okay, and then again we want to remember that the smaller units are 1/100ths of a mil. So total volume from the tip all the way up to the zero is 1 mil. 10 times 0.1 mil is 1 mil, and the smaller units are 1/100th mil. Okay. To use these pipettes, we never mouth pipette. In the old days, we used to use our mouth to create the vacuum, and we now know that's not a good idea for transferring disease-causing microbes or carcinogens. So instead what we use, in a lot of labs they'll have automatic pipette pumps, but we have these lower tech plastic pipette pumps. So here we have a smaller pipette pump, and we'll see that there's a knob here that if we rotate, you'll see that the little plunger is moving. Right? So that's how we're going to create a vacuum. The opposite end we'll see here. This is the end into which we'll put the blunt end of our serological pipette. So we want to place it and gently twist with a little bit of pressure. We want to make sure that we have a good seal. Okay. So then if we're going to create a vacuum to draw up fluid, again what we're going to do is we'll rotate this knob. We will have immersed the tip of our serological pipette into a liquid. We'll rotate this knob. That will create the vacuum that will help draw up fluid into the pipette. And then when we want to dispense or deliver the fluid, we can reverse the direction that we rotate the knob, or if we want to deliver it all at once, we can simply press down on the plunger. Okay. We'll just do one more pipette, serological pipette. So here we have a larger pipette, and we want to ask ourselves, what's the total volume? So again, the information is here at the blunt end. So the total volume on this pipette is 10 mils. The smallest units are 1/10 mils. So let's see if we can't kind of walk through this pipette here. Okay. So we can use this. This might be an example, say, on a lab practical. So we might ask you, what's the volume if you drew up fluid from the very tip up to where this tape arrow is? All right. So we can count. We want to include the volume at the very tip. So this would be 1 mil, 2 mils, 3 mils, 4 mils, 4 mils. And we want to remember that the smaller units are 1/10 mils. So 4 mils, 0.1, 2, 3, 4, 5. So 4.5 mils would be our answer. When we have our discussion of sterile aseptic transfer techniques, when we discuss making gram stains, we will walk over the use of the Bunsen burner. There's real important Bunsen burner safety rules. First of all, we never leave a Bunsen burner lit and walk away, even if it's for a few seconds. We always turn our Bunsen burners off unless we're sitting right in front of the lit Bunsen burner. We want to keep the Bunsen burner flames low to reduce the chance of someone being burnt. When we are going to light our Bunsen burners, we make sure that our bench is clear. We want to make sure that if we have long hair, that we tie back our long hair. If we have loose sleeves, we want to use rubber bands to tighten up the sleeves to make sure that we don't catch the Bunsen burner. And we also want to check to make sure before we leave lab that we've turned our Bunsen burners off. We don't want to leave the Bunsen burners on. And we'll have a demonstration of how to light the Bunsen burners. So I think for now we'll close this little video, and we'll look forward to seeing you in lab, and we'll become familiar with using these tools, hopefully to enjoy our experience in the microbiology lab.